So you may have heard of the concept of a trait skipping a generation. Well, sometimes it actually is true. The trait of white flower petals skipped the F1 generation, and it popped up again during the F2 generation. So he said purple is dominant, right? But then he says, okay, white, it disappears into the background. It recedes into the background, but then it comes back in later generations. He said white is recessive. Recessive. He didn't say it was dominant and inferior, dominant and submissive. He said dominant and recessive. The trait that shows itself in hybrids is your dominant trait. The one that comes back in later generations is your recessive trait. And as it turns out, lots of different genetic traits follow this kind of simple inheritance pattern. Not all of them. Sometimes you do see a blending of characteristics. Sometimes other types of effects come in. Sometimes you might have more than one gene that's responsible and you get some more interesting results. But a lot of traits do follow this simple dominant recessive relationship here. Now, sometimes for genetics questions, I'm going to ask you phenotypic ratio. Now, just a moment ago, I told you what the phenotypic ratio was, but I'm going to show you where that comes from, okay? Purple is a phenotype. Purple and purple. White is a phenotype, okay? So when I ask you for the phenotypic ratio, I'm asking how many of one phenotype exists compared to other phenotypes. So in this case, it would be 3 purple to 1 white. Now, when you're doing ratios, you reduce it into its smallest terms, which this already is. If you have one of your numbers as a 1, you're already in the smallest terms possible. But if I had 12 purple, and I had 4 offspring that were white, well, 12 to 4 reduces down to 6 to 2, which reduces down to 3 to 1. So you always want to reduce down to the smallest possible ratio when you're doing these kind of problems. So Gregor didn't just do this with one set of pea flowers. He did this several thousand times. If you look at his original tables of research, he's looking at not 12 purple flowers and 4 whites. He's looking at something like 6,000 purple flowers and 2,000 white flowers. It's a pretty substantial set of data. And from the mathematics, he realized that something was going on, that somehow this F1 generation remembers that it has white flower petals in its family somewhere. There's 2 pieces of information. There's one piece of information which is determining what it looks like, but there's also this other hidden piece of information from the white flower petals. So he decided to codify this concept of different types of information and say that for every trait, an organism will have 2 alleles. Very important word to know, alleles. They can have either the dominant allele, which I'll give a capital letter, or the recessive allele, which I'll give a lowercase letter. And everything has 2. And because we bred out all of the white flower characteristics from this purple family line, we're going to assume that all of these P generation purple flowers have 2 copies of the dominant allele. And all of these white flowers in the P generation have 2 copies of the recessive allele. You always use the same letter for the same kind of gene, but dominant is given in uppercase and recessive is given a lowercase. Each of these parents gives one and only one of its alleles to the offspring. Now, it's pretty simple here because these purple flowers only have a capital P to give. No matter which of these, 50% odds that they're going to get this one, 50% odds they're going to give this one. But no matter what, some capital letter P is going to be given to these offspring. Whereas the white flowers, they only have lowercase recessive alleles to give to their offspring. So that means that the entire F1 generation is going to have a mixture of these alleles. They'll be a hybrid. They'll be what we call heterozygous. Okay. Hetero means different, so we have different alleles here. Now, if you have one dominant allele, you get the dominant phenotype. You end up with purple flowers. So since all of the F1 generation got at least one dominant allele from their purple flowered parents, that means that they are going to have the purple flowered phenotype. But when we mixed two F1, when we crossbred two F1 generation flowers, two of these hybrids together, suddenly we had a situation where plants could get two of these recessive alleles. So that means that there was a 25% chance that we would get these white flower petaled individuals. Okay? We can look at that in a little bit more detail. So Gregor's work kind of went unknown for a very long time. He published in German and wasn't. Apparently that's not what people in this field were reading at the time, so he went into kind of obscurity. He later stopped doing so many experiments. He became in charge of the Abbey, so he started focusing on that kind of work, and later people would ask him about his experiments, and he said, oh, it's just a neat little trick that I do that takes a very long time to explain. So he kind of backed away from it because no one was really reading his work. 30 years later, though, a couple of guys started finding his papers and realized that they were brilliant. Hugo de Vries was one of them. Another guy was named Reginald Punnett. And Punnett said, this work is fantastic, but I think all of these tables of equations and things and charts of data, it's really difficult to explain to someone why this is working so neatly. So what if we simplified it and create a more visual method, which we call now, after Reginald Punnett, the Punnett square. So basically, here's our F1 generation purple flowers, right? They have one dominant allele and one recessive allele. Up here, we have the female's alleles. Now, both of these parents have the same dominant recessive allele set up, so both sides of this square are going to look the same. So we have one dominant here, one recessive here. For the male, one dominant, one recessive. And it doesn't matter which side of the square the males or female are on, the math will work out exactly the same. If we go down the column and we deliver this dominant allele to every square in this column, we will find, there we go, we got two dominant alleles going down, and then go across this row for the second letter, we get a dominant allele in every square in this row. If we go down this way, recessive allele and recessive allele. Go across this way, recessive allele and recessive allele. These squares represent the four possible kinds of offspring that these two parents could possibly have. Unlike with the P generation going into the F1, there's actually more than one kind of offspring that you can end up with. You could have a homozygous dominant individual, someone with two capital P's. You could have a heterozygous individual, offspring here. We have two different ways that we made that happen. Or you can have a homozygous recessive individual. You could have a situation where they get 2 recessive alleles. Now, if you count up how many of each kind you have, you get one way to make a homozygous dominant individual, 2 ways to make a heterozygous individual, and 1 way to make a homozygous recessive individual. This is a genotypic ratio. Okay. In a normal monohybrid cross, that is, we're taking 2 hybrids and we're breeding them together, you'll get a 1 to 2 to 1 genotypic ratio. We can also look at the phenotypic ratio. The phenotypic ratio, make some room here. The phenotypic ratio, we're just looking at the results, the physical results here. So we have 3 purple flowers, 3 purple, 2 1 white flower-petaled pea plant. This is your phenotypic ratio. So, when you're doing genetics problems, there's a couple of different things I might ask for. I might say, what is the genotype of this particular plant? Genotype, you're going to tell me what alleles are there, what the actual genes present are, either with a combination of upper and lowercase letters, or you might say something like homozygous dominant. If I ask you what the phenotype is, I'm asking what they look like, right? You would say it's a white flower petal, it's a purple flower petal. If I ask you for the genotypic ratio, I'm asking you how many of each genotype there are compared to the other genotypes. If I ask you for the phenotypic ratio, I'm asking you how many of each phenotype there is compared to the other phenotypes. I'm going to say this part probably several times throughout these recordings, but if you have an organism with the recessive phenotype, they must have the homozygous recessive genotype. That means they have 2 copies of the recessive allele. If you have the recessive phenotype, you have 2 copies of the recessive allele. Okay. So here is the same experiment, but we're looking at the phenotypic and genotypic ratios. This plant here is homozygous dominant. Homozygous, homo means same, homozygous means that it has 2 copies of the same allele. This plant down here is homozygous as well, but it's homozygous recessive. All right. These two, it doesn't matter which parent gave them the dominant or the recessive allele. What matters is that they have 2 different copies, that they're hybrids here. So, we have this slide is wrong. We have 2 different ways to make heterozygotes, 1 homozygous dominant and 1 homozygous recessive, giving us a genotypic ratio of 1 to 2 to 1. For phenotypes, we have 3 purple flowers and 1 white flower, so the phenotypic ratio is 3 to 1. Now, it's very common for students to want to say something like, oh, there's 4 plants here, one of them is white, so it's 4 to 1. But that's wrong. That's you're basically making a fraction at that place. You're saying 1/4 of the plants is white. If I'm asking you for a ratio, it's how many of each kind of thing is there, and you want to reduce it the same as you would a fraction, so you put in the least possible terms. It turns out that this relationship is not limited to just flower color, that there's many different traits in peas and in humans and other animals and other organisms as well that follow this dominant recessive pattern. So with pea plants, Gregor checked this with flower color. He checked this with the plant height. He checked it with the color of the peas, yellow peas versus green peas. As it turns out, green peas are a recessive trait. He checked it with the shape of the peas. It turns out wrinkled peas is recessive, whereas nice big round peas is dominant. He checked it with the color of the seed pod, which as it turns out is independent from the color of the peas. You can have green seed pods with yellow peas or yellow pea pods with green peas. He checked it against the pod shape, whether it was inflated or constricted. Constricted looking like it's wearing a very, very tight coat, so you can see all the little peas inside. And with the position of the flowers on the plant itself, whether it grew out of the axial position, the side of the vine, or the terminal position, the very, very top of the design. In each of these cases, he found the same 3-to-1 phenotypic ratio popping up in the offspring. Okay. So we now know that there are alternate versions of genes which ride on the chromosomes, which we call alleles. Now, Mendel didn't know anything about chromosomes. It hadn't been invented yet. It hadn't been discovered yet. In fact, if you take a look at when he published his work versus when DNA was discovered by a gentleman named Friedrich Miescher about 6 years after Mendel discovered his pea plants. And Miescher was actually over in the University of Tübingen, which is today about a 6.5-hour drive away from the monastery where Gregor Mendel was. So this work was happening at nearly the exact same time, but Mendel did publish first. So he had no idea that nucleic acid even existed, let alone what its role in inheritance was.